Hey guys, today we're going to cover chapter 3, and this is the notes that I have that are the consolidated notes. I know that chapter 3 is a huge chapter. It has over 200 slides. I've shortened it down a little bit. I'm actually going to chunk this lecture out. I'll stitch them all back together for you, so if there's a little bit of lack of continuity, that's why. So we're going to start off talking about cells. They are the living units of life. It is the smallest unit that is capable of exhibiting all the characteristics of life. So we're going to start off with the cell theory. So the cell theory was developed by a bunch of scientists that basically said in order for a cell to be a cell, these are the things it has to do, and these are the things we know to be true of cells. So a cell is a structural and functional unit for life. It is the smallest thing capable of supporting all of life's functions. The structure and function are complementary. So like we said, that the structure dictates the function. This is a common theme in anatomy and physiology. Very true here. We have cells that have a large variety of shapes, sizes, and functions as well. And down at the bottom there, it talks about how cells come from existing cells. So we have a long time ago disproved the theory of spontaneous generation, which says that cells essentially just like sparked out of thin air. So we know that cells come from other cells. There had to be one cell in order to create two cells and so on through like mitotic division and other types of budding and asexual reproduction as well. So as I said, we have a lot of different cellular types. So we have a large diversity among our cells. So there's over 200 different types of just human cells. They differ in the sizes, the shapes, the components that are within each of the cells. Here's some pictures of them for you. So these are all cells that you'd find inside of a human, right? They all look very different. And remember that the structure dictates the function. So the way that it looks kind of gives you a hint as to what its job might be in the body. So like for A, these are cells that connect body parts, form linings, or transport gases. So, like your erythrocytes, which are your red blood cells, are like a little donut shape with that divot in the middle. That's where the heme group lies to hold oxygen and deliver that to the different parts of your body, right? So it's kind of like a little bucket shape to hold on to all those extra molecules. So, like I said, the structure dictates the function, and you can read the others on here for yourselves. So cells are our living units. So a generalized cell, all cells come from have common structures and common functions. So regardless of what kind of cell you're talking about, there are similarities between them. So human cells have three basic components. That's true for all human cells. Now remember, we have a whole bunch of different types, right? So these are things that we know to be true for all human cells. They all contain plasma membranes, which is also called a phospholipid bilayer or a cellular membrane. This is a flexible outer boundary, kind of like a water balloon on the outside of a, water balloon. We also have cytoplasm. Cytoplasm is like the empty gel-like solution that exists between the nucleus and the plasma membrane. It kind of like surrounds all of the organelles and cushions them. We also have a nucleus because humans are eukaryotic organisms. So our cells are called eukaryotic cells because they contain a nucleus. And of course, the nucleus's job is to contain the DNA. It is the control center because, of course, your DNA is the blueprint to create you. Here's a picture of what our eukaryotic cells look like. You can tell by looking at this that it is a highly structured cell. So this is very different from like our prokaryotic cells, like our bacterial cells that we've learned about. So our eukaryotic cells are highly structured, highly compartmentalized. And so here you can see all the different color coding of all the different organelles. And we will go into that further later in this lecture. So we have our intracellular material, which you can say here. So intra, inside. You also have extracellular material, which is outside of the cell. So some of that can be the surroundings of the cell, or it can also be like projections from the cell itself. So these are all substances found outside of cells. So you have different classes of extracellular material, and here's some of the ones we're going to be talking about. So you have extracellular fluids, like body fluids, if we're talking about, the human body, because this is A and P, such as our interstitial fluids, which are when we have cells submerged or bathed in a fluid. We have blood plasma, which is the fluid part of your blood. You have cerebrospinal fluid, which is the fluid surrounding your nervous system organs. Next, you have the cellular secretions such as saliva and mucus. And you also have extracellular matrix, which is the substance that acts as glue to hold cells together. So cellular transport is going to be the first big topic. So that was a little bit about just like cells in general. Cellular transport is the first big topic of chapter 3. There's 4 big topics. This is going to be our first one. So the plasma membrane is of huge importance when we're talking about transport, because this is how cells are able to move things in and out. And of course, you want to bring in the things that you need, like, food sources, water, other nutrients, things like that, and get rid of things that you don't need, like waste products or things that you have in excess that could be toxic to the cell over time, right? So our plasma membrane, our cellular membrane, is something that is selectively permeable. It acts as a barrier. It's actually an active barrier that's going to be separating the intracellular fluid from the extracellular fluid. So it plays a dynamic role in controlling what leaves and enters our cells, because it's constantly moving. It is a fluid mosaic model of our cellular membrane, because things are always moving. It is not just this stagnant, still thing that just surrounds the outside of our cells. It is constantly in motion. It's constantly bending and flexing and doing all of the jobs that it needs to in order to keep our cells in homeostasis. So let's talk about the structure of this membrane. This membrane is a phospholipid bilayer. Of course, bi meaning two. So phospholipids are a type of fat that has a phosphate group on the head. This allows them to have both hydrophobic and hydrophilic regions, which we'll talk about in a minute. But we have phospholipid bilayers on the outside of all of our cells. And this is true for every single cell that's ever been a cell and ever will be a cell. They all have to have a plasma membrane, also called a phospholipid bilayer. We also have specialized membrane proteins that are kind of scattered throughout this membrane. We have some that are transmembrane spanning, so they allow things to pass through. We have some that act as like receptors and can catch things that are in the extracellular matrix around our cells. As it says here, though, we have a fluid mosaic model for our current plasma membrane, because all of these pieces, whether it's the phospholipids themselves or the proteins or even carbohydrates and other lipids that are also associated with the membrane, are constantly in motion, and it's made up of many different pieces. It's not just this uniform structure. The surface sugars that are on the outside of our cells are called the glycocalyx. It's a whole bunch of different sugars that are interacting to typically provide cell-to-cell communication between our cells. And we also have membrane structures that help to hold our cells together through cell junctions. And there's 3 different types of junctions that animal cells exhibit that we're going to talk about soon. So this is kind of a picture for you to show you what the phospholipid bilayer looks like. So it explains the functions of the plasma membrane here, that it's a mechanical barrier, it's a selectively permeable barrier. There's an electrochemical gradient to allow things to go in and out according to their concentration gradient, and so on. So stop, look at this picture, write down these functions of the cell membrane, as well as like these little labeling, the little labels that are on the outside and the inside of the structure here, because you will need to know these for your exams. So make sure that you understand where everything is located, and later also what they do, why they're there. So our membrane lipids, there's different types of lipids that we have that are making up this giant structure here called the phospholipid bilayer. So we have 75% of these lipids being phospholipids, which are 2 parts. It's a phosphate head and a fatty acid tail. So the phosphate head is polar. It's a charged part. So they want to interact with water, because remember, like dissolves like, they like to, both polar materials will want to interact with each other. So those are hydrophilic or water-loving. You also have the fatty acid tails, which remember are just hydrocarbon chains, which are hydrophobic, and they are nonpolar. There is no charge. They are water-fearing or water-hating. They cannot interact with water. Our next 5% is going to be glycolipids. Glyco is a prefix meaning sugar, and lipids, of course, are fats. So this is lipids with sugar groups on outer membrane surfaces, which could help with like cell-to-cell communication or recognition. We also have 20% of the lipid bilayer being made of cholesterol. Now, remember, cholesterol is a steroid, but it's also considered a lipid class. And this increases our membrane stability. It also increases membrane fluidity, because they're these large 4-ring structures that are embedded in between our phospholipids, and this helps to maintain the fluidity of the membrane to prevent them from being rigid. And if you were an organism living in a very cold environment, you'd have a very high concentration of cholesterol in your cellular membrane in order to keep your phospholipid bilayer fluid at those cold temperatures. Next, we'll talk about our proteins. We just went through our lipids. So our proteins, they serve a whole bunch of different functions. We're going to talk about a couple of them today. So our proteins allow for cell communication with the environment. So they act as receptors on the outside to, receive messages from the outside world, whether it's from another cell or from just other signals that are present in extracellular environments. They also make up about half the mass of the plasma membrane. Like I said earlier, they can be embedded in or just kind of like partially stuck into the membrane to serve different functions. Most of them have a very specialized function within the membrane. Some of them are free floating and they just kind of like stay in close association with the phosphate heads on either side of the membrane. Some of them are tethered and actually inside and embedded into the structure of the membrane itself. There are two different types we're going to talk about. We have integral or integral proteins and peripheral proteins. So our integral proteins are integrated. They are firmly inserted into the membrane. Most are transmembrane proteins, which means that they're going to completely span the membrane. So they're present on both the internal and external surfaces of our cellular membrane, and they're usually used to transport things if that's the case. So they have both a hydrophobic and a hydrophilic region. Of course, the hydrophobic region is going to be interacting with the hydrocarbon lipid tails, and the hydrophilic region is going to be interacting with the water near the polar heads on either side of the membrane. They function in transport, as I said, because they are kind of like channels. They're gateways that exist between the membrane. So it's on both ends of the membrane, so things can easily pass through. Of course, there are certain things that they do not allow to pass through, but then they can also function to be enzymes or receptors to carry signals to the interior or exterior surfaces of the cell. Next, we have our peripheral proteins. So peripheral proteins are on the periphery or towards the outside. So this could be either the outside of the cell or the inside of the cell. It's just the outside of the cell membrane itself. So they are loosely attached to integral proteins. They include filaments on intracellular surface that are used for the plasma membrane support. And the intracellular filaments here are referred to as the cytoskeleton, which we'll talk about in the second part of this. Our peripheral proteins function as enzymes, typically also as motor proteins to help shape and move things around the cell, and then also in cell-to-cell communication and connections. So as I said, proteins have a whole lot of responsibilities, a whole lot of functions. And again, their shapes are going to help give a little bit of insight as to what their functions may be. So these are a few of them. I'm going to actually let this be something that you're going to read on your own. But you have transportation capabilities. They can act as receptors for signal transduction. They can attach to the cytoskeleton and to help with support and structure of our cells, enzymatic activity, intracellular joining, and cell-to-cell recognition and communication. Next, we're going to talk about our glycocalyx. So previously, we've talked about our lipids, we've talked about our proteins, and then, of course, our next major class is going to be our carbohydrates here. Notice that I'm not going to talk about nucleic acids because nucleic acids do not exist in the plasma membrane, because where are nucleic acids? That's correct. They are inside of the nucleus of our cells, of course. So the glycocalyx is referring to sugars. Glyco, again, is a prefix meaning sugar. And so our sugars are carbohydrates. So these are oftentimes sticking on the outside of our cellular surfaces. So some of these sugars actually attach to lipids, which are called glyco, meaning sugar, lipids, fats. And some of them are attached to proteins. So they would be glyco, meaning sugar, proteins, for protein, right? So every cell has different types of patterns and sugar coating around the outside, because each cell type that has a different function is going to have a slightly different structure. So the glycocalyx is going to be slightly altered depending on the type of cell and the function of that cell. This sugar coating functions as specific biological markers for cell-to-cell communication, and it's very important. It allows our immune system to recognize our self cells from our, quote, non-self cells. So this is like your white blood cells can identify, oh, this is supposed to be here, this is part of my person, whereas an invader, like a bacterial cell, would not have the correct, quote, correct biological marker on the outside, and it would signal that this is a foreign invader, and then can be marked for destruction to help keep you healthy. Next, we're going to talk about cell junctions. So this is how we have multiple cells kind of like joining together. So some of our cells are, quote, free. They're not bound to other cells. So some examples would be like blood cells and sperm cells. These are cells that are typically, on the move. They're not going to be bound together to form like tissues, which are going to make up our organs and so on, right? These are cells that kind of just have their specific function. They're going to do their own thing. It's not like you have blood tissue. You just have blood, which is a moving thing, right? Most cells, however, are bound together to form tissues, and tissues make up our organs make up our organ systems, and so on and so on. Great. Okay, so there are three ways that cells can be bound to each other when we're talking about human cells. So we have tight junctions, we have desmosomes, and we have gap junctions. So let's look at those. Our tight junctions We're dealing with integral proteins on adjacent cells, so two cells that are next to each other, that are going to fuse and form an impermeable junction that encircles the whole cell. So this is there to prevent fluids and most molecules from moving in between the cells. So they're just keeping them, it's closing the gap between them, essentially, an impermeable membrane, essentially. So where might this be useful in the body? We have a high concentration of these tight junctions in our epithelia to help prevent movement between cells. So we might see this in the nephrons of our kidneys. We might see these in bile ducts, like in the liver, and so on. Here's a picture of what this actually looks like. So our tight junctions, again, are impermeable junctions that form continuous seals around the cells to prevent molecules from passing through to the intracellular, the inside space of our cells. Our next type of cell junction is called a desmosome, or desmosomes for plural. So these are cell junctions that formed when linker proteins that are called cadherins of neighboring cells actually like interlock, kind of like imagine like a zipper, like you're zipping up and down your jacket or something. They actually physically interlock with each other to create kind of this zipper between the cells. So the linker protein is anchored to its cell through a thickened button-like area on the inside of the plasma membrane, and these are called plaques. We'll look at this in a minute. It won't feel so wordy. Okay, then we also have keratin filaments that connect the plaques intracellularly for added anchoring strength. So this is just, keratin is a protein, so it's going to act for structural strength. And our desmosomes allow some give between cells, and it reduces the possibility of tearing under tension. So we might see these in areas that experience intense mechanical stress, like our cardiac muscles. If they stop, you're dead, right? So they're constantly under tension, they're constantly under stress. Same thing with your bladder. It's constantly inflating and deflating, so it's constantly in motion. Your GI mucosa of your gastrointestinal tract, right? All of these things, like where your digested food and, feces is moving through your body, it's in constant motion. And those involuntary muscle contractions are called peristalsis. So here's a picture of our desmosomes. So like I said, it's kind of like a zipper. You have your little linker proteins that are attached to a plaque on the intracellular side. These are anchoring junctions that bind adjacent cells together that act as kind of like a Velcro or a zipper to help form an internal tension-reducing network of our fibers here. Lastly, we have these things called gap junctions. So these are transmembrane proteins called connexons that are going to form tunnels that allow small things to pass from cell to cell. So it's a gap. It's like kind of like a little hole that exists between one cell to the next. That way they can allow for quick and easy transport of materials. So these are used to spread ions, simple sugars, and other small molecules between the cells, as I just said. It allows the electrical signals to also be passed quickly from one cell to the next cell. So if you're like receiving like from the nervous system an electrical signal or something to have some sort of response, this would allow them to perpetuate faster if you have gap junctions. And these are used in cardiac and smooth muscle cells. And this is a picture of what that would look like. So we have our gap junctions. They're communicating junctions that allow ions and small molecules to pass through from one cell to the next, and are particularly important for communication in the heart cells and embryonic cells as well. Okay, so we have all these different types of junctions. We have all of these, characteristics of our membrane, but how do things actually move in and out, right? It is selectively permeable. That's great. We've kind of talked about the structure. So you know what it looks like, the functions of all these, interworking pieces, but how is it selectively permeable? So some molecules are able to pass through easily and some are not. So there's two different ways that substances can actually cross through the plasma membrane. The two processes are passive transport and active transport. So passive is no energy required, whereas active transport requires ATP. Literally the letters A, T, and P are all part of that. So passive is when there is no energy. There are two main types of passive transport called diffusion. So we have simple diffusion. You also have facilitated diffusion and osmosis. So osmosis is specifically the diffusion of water molecules. So that one is very special. Simple diffusion would be like someone sprays perfume on one end of a room and then slowly you can smell it all the way in the back of the room. The molecules move from a very high concentrated area to an area of very low concentration passively without energy. So like I said, with osmosis, that would be true, but just with water. And where we have our carrier and channel mediated facilitated diffusion is just means assisted diffusion. So this is simple diffusion that's occurring with the assistance of some sort of transmembrane protein, typically a carrier protein or a channel protein. There's no ATP required here, but there is a helper that needs to be there, and that's why it is called facilitated, which literally means aided or helped diffusion. It's just passive diffusion that occurs through one of these specific proteins. And the second type is called filtration, which is a type of transport that usually occurs across capillary walls. So in diffusion, so we have collisions between molecules in an area of high concentration that cause them to kind of bounce around and be scattered away to a less concentrated area. So especially like this is easy to imagine like gases, like gases the molecules are moving very quickly. They're constantly having all of these collisions. And as they bump into each other, they're going to be like ricocheting and like bouncing away from each other, right? So that's what causes a movement from high concentration to low concentration. The difference between high to low concentration is actually called the concentration gradient. If you imagine somebody's like, it used to be cool to have like ombre hair, no shade to the people that still have it, but it used to be cool to have ombre hair. So you have like a high concentration of color, usually at either like the top of your hair, the bottom of your hair, and then it slowly fades, right? So it's a concentration gradient. Okay. Diffusion is the movement of molecules down their concentration gradient. So molecules down their concentration gradient from an area of high concentration to an area of low concentration is considered down the gradient, whereas up or against the gradient would be from an area of low concentration to an area of high concentration, because they don't naturally go in that direction, which is actually the opposite type of transport called active transport. So again, in diffusion, moving with the concentration gradient or down the concentration gradient from an area of high concentration to an area of low concentration, no energy is required. And the speed of diffusion is influenced by the size of the molecules as well as temperatures. You know that as you increase the temperature, the molecules are going to be moving faster, which means that the rate of diffusion will be. So this is an example of diffusion happening with a dye pellet inside of water. You can see that you have a very high concentration of this dye in the pellet, whereas at the extremes, at the outsides of the beaker there, you're not going to have any of that. It's just H2O probably. Diffusion is going to occur, so you slowly see the molecules moving from the high concentration out towards the low concentration until there is an equilibrium that is reached when the dye is evenly distributed amongst the entire beaker. So molecules have natural drive to diffuse down their concentration gradients that exist between extracellular and intracellular areas. So things can move across the membrane based on their concentration gradient. And diffusion says that they can do it without the use of energy. So plasma membranes stop diffusion and create concentration gradients by acting as selectively permeable membranes. So if some of these molecules are small and non-charged, they can typically just diffuse straight through the membrane, whereas if they're a little bit bigger, they're going to need things like facilitated diffusion to use our transmembrane proteins to get them from one side to the other. Our nonpolar hydrophobic lipid core of the plasma membrane blocks diffusion of, most molecules, because a lot of the ones that are present in the body are actually charged, so that would make them polar. Okay, so our molecules that are able to passively diffuse through the membrane include things that are lipid soluble and nonpolar substances. You have very small molecules that can pass through the membrane or membrane channels, things like gases. Okay, then you also have larger molecules that are assisted by carrier molecules. This would be an example of facilitated diffusion because they are too large to pass straight through the membrane itself. So again, simple diffusion, we have our nonpolar lipid-soluble hydrophobic substances that are able to go directly through the phospholipid bilayer. Like I said, this is like gases. We have oxygen, carbon dioxide, fat-soluble vitamins as well. Sure of what that would look like. On the extracellular side, you can see that you have a high concentration of these lipid-soluble solutes, and then you have the blue plasma membrane in the middle of the screen, and these little molecules are diffusing straight through simple diffusion through our phospholipid bilayer to reach the cytoplasm on the intracellular side of the membrane. We also have something called facilitated diffusion, which is when we have hydrophobic molecules that are a bit larger, such as like glucose, so like monosaccharides, amino acids, ions, which are charged. They're transported passively down their concentration gradient by either carrier-mediated facilitated diffusion or channel-mediated. So either we have substances that are binding to protein carriers in a carrier-mediated diffusion, or you have channels that allow substances to move through water-filled channels in order to get to the other side of the membrane. So carrier-mediated means that you actually have something binding to the carrier protein, whereas in a channel-mediated, there's no binding of the molecule. It's literally just using it like a slip and slide because it's full of water and it's just able to go in and out of the cell. So in carrier-mediated facilitated diffusion, we have carriers that are transmembrane integral proteins. So transmembrane means that they are present on both the intracellular and extracellular surfaces of the cell membrane. So these carriers transport specific polar molecules, such as sugars, amino acids. These are things that are too large for simple diffusion. They're too large to just go straight through the membrane, and they're too large for our membrane channels as well. So an example could be a glucose carrier that will only carry glucose molecules and nothing else. They have a very high specificity typically. The binding of the molecule causes the carrier to change shape, which moves the molecule in the process. So we'll look at that in just a second. It's literally a conformation change that happens with the protein that's embedded in the plasma membrane. The binding is limited by the number of carriers present. So if the carriers are saturated, this means that all of the areas that they're able to bind have been filled with one of the transporters that's about to go through. So here's an example of that. On the outside, the blue side, you see that you have these lipid-insoluble solutes such as sugars, amino acids, so on. Once they enter into the carrier protein, there's a conformation change, which makes it flip to the other side of the membrane. So the shape change is actually going to release the solute to the intracellular side of the membrane. So again, this is called a carrier protein, and it's specific for one specific chemical, as most proteins are, they display high specificity. So this is how they work. There's actually a conformation change that happens with the protein. It changes its shape in order to move things from one side of the membrane to the other. Notice there's still no ATP used here. We also have channel-mediated facilitated diffusion. So channels, like I said, is kind of like a slip and slide. They're filled with aqueous solutions, so water-containing solutions, that are formed by our transmembrane proteins. So these channels transport molecules such as ions or water, like larger amounts of water, like through osmosis, down their concentration gradient. Now remember, this is still passive diffusion or passive transport, so everything that we're talking about right now is down the concentration gradient from an area of high concentration to an area of low concentration. We have a specificity based on the pore size and or charge of the molecule, so certain channels might only move certain products or certain substances. And our water channels that are specific for transporting larger amounts of water are called aquaporins. So that's like water pores, essentially. We have two different types of channel-mediated facilitated diffusion. We have leakage channels and we have gated channels. So leakage channels, think about like things that are leaking, right? It's not very regulated. They're always open. Whereas our gated channels, think about like a gated community, right? They're controlled in some way. So these are controlled by chemical or electrical signals and or gradients. So here's an example of what our channel looks like. It's a little bit different from this guy right here. There's no conformational change. It's literally just a slip and slide. It's full of water and it's able to move these molecules from the exterior to the interior because that's where we have our high to low concentration. Can they go in the other direction? Yeah, absolutely. But it depends on the concentration of the molecule that is moving. We also have osmosis. Osmosis is diffusion, but it is specifically the diffusion of water and only water. So this is the movement of a solvent, like water, across a selectively permeable membrane. So it's not just like, oh, I put water from this cup to that cup. That's the movement of water. Okay, no. So osmosis has to be through a selectively permeable membrane. So water diffuses through plasma membranes. So through the lipid bilayer, they often use water-specific channels, also called aquaporins, that we just talked about a second ago. And then the flow occurs when water concentration is different on the two sides of the membrane. So that's when we get like osmolarity. We have a difference in the amount of water that's on the interior or the exterior of the cell. So here's an example of water, those little H2O, little Mickey Mouse looking guys floating around. You can see that water is coming in, and you see there's two different options here. You have water that's coming in through an aquaporin, as well as water coming through just the phospholipid bilayer. Both can happen, but typically water moves through aquaporins. So that's what's happening in this image. So I said this word earlier, osmolarity. It's the measure of the total concentration of solute particles. So water concentration varies with the number of solute particles because solute particles displace water molecules. So essentially you have water and then you have stuff in it. The stuff is called a solute. So wherever you have more solute, you therefore have less water because of displacement. Okay. So when solute concentration goes up, water concentration goes down and vice versa. So that's just another way to say what I just said. So basically water is going to be moving through osmosis, which is just the diffusion of water across selectively permeable membranes, from areas of low solute, which means high water. If you have not a lot of stuff, you have a lot of water concentration, to areas of high solute, which is low water concentration. So I still think that it's high to low concentration, because if you're just talking about water, because osmosis is referring to water, so if it's a high concentration of water then to a low concentration of water, it still follows that principle, but it's flipped if you think about it from the solute point of view, and I'll give you some examples of this in class. So when solutions of different osmolarity are separated by a membrane that's permeable to all of the molecules, both the solutes and the water can actually cross the membrane until there's a dynamic equilibrium that's reached. Now understand that equilibrium doesn't mean that the molecules stop moving. It just means that they're moving at a net of zero. So they're coming in and out at the same rate where there is no change in the concentration at any given time. So like I said, that's our definition of equilibrium. You have the same concentration of solutes and water molecules on both sides of the cell, and understand just because they've reached equilibrium, there is still motion happening in and out of the cells, but it's happening at the same rate, so as the concentration is never affected. These are called U-tubes, and this is something that's very common to demonstrate where water or solutes are moving. So here we have a membrane permeable to both the solutes and the water. You'll notice that the water and the solutes are moving at the same rate here. So there's not an influx of water on one side to raise the level of water on one side, but what you see is that you have solutes and water both moving in order to, down their concentration gradients in order to reach an equilibrium here. So when solutions of different osmolarity are separated by a membrane that is permeable not only to water and not to the solutes, this is only dealing with osmosis. So osmosis is going to occur until we reach an equilibrium. So this is going to look a little bit different because our solutes are not moving this time around. So here we see an influx of water that's moving from the left to the right. You can see in the U-tube, you have more water now on the right side than you did initially. Essentially, what you're doing is you're diluting all those solutes because the solutes cannot move in this scenario. The membrane is not permeable to the solutes. So here you just have the movement of water, which is called osmosis, and you see an influx of water. It looks like it's going against gravity here, but this would actually happen because water is going to be moving from an area of high concentration to an area of low concentration. And again, it's the opposite if you're considering it from the solute point of view. So our movement of water actually causes these different pressures to occur. So we have hydrostatic pressure, which is the pressure of water inside the cells that's going to be pushing out on the membrane. And we have osmotic pressure, which is the tendency of water to move into a cell by osmosis. Okay, so more solutes inside of a cell means that you have higher osmotic pressure. So a living cell has a limit to how much water it can actually hold. So like, yes, the gallon challenge, you can drink too much water and actually essentially drown yourself and die. Water can also leave our cells, which causes cells to shrink. So if you become severely dehydrated, you don't have enough water, your cells will physically shrink. Okay. So the change in the volume can disrupt our cell function. You're changing the structure, which will therefore change the function. This is again our overarching theme of the whole class, especially in our neurons. So this is called tonicity when we're talking about how the cell is influenced based on the type of solution that it's in. So the ability of a solution to change the shape or tone of cells by altering the cell's internal water volume. We have different solutions here. We have an isotonic, and in freshman year, I very grammatically correct told you I so healthy, isotonic I so healthy. Yes, that's not correct grammatically, but it makes sense because this is the solution that cells are happiest in. So it's the same osmolarity on the inside and outside of the cell. So there is no change in volume. The cell is exactly as it should be, carrying out its functions, doing its little job, and living its little happy life. Isotonic, I so healthy. Hypertonic, hyper, R, wrinkled like a raisin. Hyper, if you give a kid a lot of sugar, they are hyper, higher, okay? Has a higher osmolarity than the inside of the cell. So this causes water to flow out of the cell, wrinkled like a raisin. Water is going to be exiting because it's going to be wrinkled up. So this is a shrinking, okay? And that's called crenation of a cell. You also have a hippo, big fat O solutions, which means low. So it's low solute concentration. So you have water flooding into the cell, which is going to cause it to break open and lyse. Lyse means to pop or to break. Okay. So the cell is going to swell so much that it will actually break open. Here's some examples of what that would look like in our red blood cells. Isotonic, iso healthy, you have water moving in and out at the same rate. You have equilibrium arrows. In the hypertonic solution, you have water moving out, which means that you are going to have crenation happening, which is when this cell actually shrinks. Hyper, R, wrinkled like a raisin. Hypotonic, hippo, big fat O, you can see that we have a huge influx of water going into the cell, and then the cell is swelling and it will eventually burst. So we just finished talking about passive membrane transport. So now we're going to be talking about the opposite of that, which is going to be active membrane transport. So remember that passive does not require any energy, and that's like a huge difference between the two. Active transport absolutely requires energy, and usually it's energy in the form of ATP. So there's two major types of active transport that we'll talk about. You have active transport and vesicular transport. So both of these processes require ATP to move their solutes across the membrane. And these are the reasons that you might use active transport over passive transport. One, that the solute could be too large for our passive channels. The solute could also be non-lipid soluble, or the solute might not be able to move down the concentration gradient. Maybe we're trying to move it against the concentration gradient, which is another big differentiation between passive transport and active transport. While passive transport goes from high to low concentration of that solute, in active transport, it's going to go the opposite direction. So it's going to go from an area of low concentration to an area of high concentration. So that's against the way that it typically wants to flow. This is when we would need energy to move it. So active transport requires carrier proteins. They're called solute pumps or just pumps, and it's usually the name of the solute then pump, like sodium potassium pump, move sodium and potassium. So we have our solutes that are going to bind specifically and reversibly to our proteins that are going to be moving them. So they're just going to come into the protein, bind, and then that will be reversed when they're on the other side of the membrane. And you'll see pictures of that in a second. And there's two different ways that we can transport things. We can transport things in antiporters. So this is when you have one substance going into the cell while another is coming out of a cell. So this is in opposite directions, like antiparallel. They are antiporters would do this function, while symporters are going to transfer substances in the same direction. So either both substances in or both substances out of our cell. And as I said earlier, this is when we have solutes moving against their concentration gradient. So this is going to be from an area of low concentration to an area of high concentration. And because this goes against the flow of traffic, this requires ATP, which is energy. So we're going to talk about two types of active transport. You have primary active, which the energy is used directly from ATP. So when you hydrolyze ATP, you're cutting off that the terminal phosphate to create ADP. And then secondary active transport, which is going to obtain its energy indirectly from the ionic gradients that are made during primary active transport. And this will make some more sense when we look at the pictures. So primary active transport, this is when we have the energy that's hydrolyzed from our ATP. So basically you have ATP. So it's adenosine with 3 phosphates on it. When you cut off that terminal phosphate, you're going to release a lot of energy. And that terminal phosphate will attach to the protein that is doing the transportation, and it will cause a shape change in order to open up on one side of the membrane versus another. So the shape change causes the solutes to bind to the protein to then be pumped across the membrane. And then when you have the dephosphorylation, when the phosphate comes back off, is when it flips to the other side of the membrane again in order to transport the solutes from one side to the other. An example would be like our sodium potassium pump. So that's what we're going to focus on here. So a sodium potassium pump is our most well-known, most well-studied pump. It's an enzyme. You know that enzymes are catalysts that speed up chemical reactions. In this case, it's going to be speeding up the transport of sodium potassium against their concentration gradients. So this particular one is called sodium potassium ATPase. Ase tells you that it's the name of the enzyme. It's going to pump sodium out of the cell and potassium into the cell. So this is an antiporter because they're going in opposite directions. It's located in all plasma membranes, and we have a lot of them in our nerve cells and our muscle cells because they help to transport signals and then also to help you contract your muscles. So we have leakage channels that are located in the membranes. We talked about this previously. We had gated and leakage channels. So leakage channels are just going to allow like passive movement of sodium and potassium both in and out of the cell. So they're traveling down their concentration gradients in that situation with a leakage channel. So the sodium potassium pump is going to work as an antiporter that I said earlier that's going to pump sodium out of the cell and potassium back into the cell, because passive transport's letting them move the opposite directions, but we need their concentrations to be different in order to contract our muscles and to send signals through our nervous system, right? So we have to move them back against their concentration gradient. So we're going to need ATP to do that because they're going in, like the upstream or the wrong direction, essentially. And as I said, this is essential for our muscle and nerve functions. So here's a picture of kind of what's going on with our sodium potassium pumps. Pay attention to which side of the membrane the sodium potassium pump is facing. So for instance, on number 1, it's facing the internal side with the cytoplasm. This is when you're going to have our little yellow molecules here called sodium, Na+, that are going to attach to our protein, and then ATP is going to come in and it's going to donate a phosphate to the pump, which is going to make it change directions because you're phosphorylating it. And then the pump is going to change directions to the outside. So now it's open on the extracellular fluid side, and it's going to release those sodium molecules. And then you see that you have the little blue-green molecules called K+ or potassium that are going to attach to our antiporter, our protein here. And then that phosphate group comes back off, which is called dephosphorylation because you're removing it. And then our protein is going to flip back to the other side of the membrane, which you can see happening there as well. So then you can release the potassium to the internal surface. So secondary active transport is when we're not using the energy directly that's released from ATP when you cleave off that last phosphate. So secondary active transport depends on the ion gradient that's created because of the primary active transport system. So this is typically removing things that are charged molecules. So when you have a buildup of a charge on one side of a membrane, that's an ion gradient. So energy stored in gradients is used indirectly to drive transport of other solutes. So if you have low sodium concentration that's maintained inside the cell by our sodium potassium pump, it's going to strengthen the sodium's drive to want to enter the cell. And our sodium can drag other molecules along with it. So as it's being transported, it can drag other things along with it. And this is called like a symporter because it's symportation. You're putting two things that are going the same direction. So in this case, you have sodium as well as another molecule. Sometimes this could be sugars, it could be amino acids, it could be other ions that are just floating around. So all of them are going to be moving via secondary active transport. It's kind of like piggybacking. So you have your primary active transport, the ATP-driven sodium potassium pump that's storing the energy created from the concentration gradient for sodium entry back into our cell. Okay. And then in the second picture there, you see those glucose molecules. They're kind of piggybacking. So as we're bringing sodium back in, the glucose is going to be symported along with it. So as the sodium diffuses back across the membrane through a co-transporter protein, so it will transport multiple things at the same time, it drives the glucose against its concentration gradient into the cell. So again, still against its concentration gradient. It's just kind of piggybacking off of the energy that was built up in our gradient that was created in primary active transport to drive the movement of molecules in secondary active transport. So vesicular transport is also a type of active transport. So vesicles are like these little membrane-bound bubbles essentially that exist in our cells, and that's we're going to talk about the different ways that they're used. So this involves the transport of large particles like macromolecules or fluids across a membrane in a membranous sac. So they have their own membrane. They are membrane bound when they exist within our cells. And this requires cellular energy in order for this transportation to happen. So vesicular transport can include the following processes: endocytosis and exocytosis. So if you have endocytosis, it literally means into the cell. So this is when we're bringing things into the cell. And there are a couple different ways that you can endocytose something. So through phagocytosis, through pinocytosis, and receptor-mediated endocytosis. And these are all things that we'll talk about in a few minutes. Where on the flip side, exocytosis is when you're transporting things out of the cell, exit, out of the cell. And we also have here transcytosis, which is when something comes into the cell, crosses the cell, and goes through the other side of the cell. And you have vesicular trafficking to transport things from one area or one organelle to another organelle in the same cell or to another cell altogether. So endocytosis again is into, so this involves the formation of a protein-coated vesicle. So it's usually going to involve some sort of receptor and can be very selective because receptors are typically proteins, and they are very selective as to what can attach to them, kind of like a lock and key. So the substance that's being pulled in must be able to bind to the specific receptor in order to be brought in. Sometimes pathogens, which are things that can cause diseases like viruses, bacteria, stuff like that, can hijack those receptors. Like they can bind to them as well. Like they're mimicking our shapes. So then their lock and key mechanism fits together with ours. And that's how we can get viruses and bacteria into our cells. They disguise themselves as something that's helpful. So our cells are like, oh yeah, man, come on in. And then it's like, psych, I'm going to kill you. That's what happens. Okay. Once the vesicle is pulled into the cell, it can do one of two things. It can go under transcytosis, which just means it's going to travel through the cell and out the other side, or it can fuse with a lysosome. And remember that the lysosome is like the little stomach of a cell. It's going to digest that vesicle because it's full of hydrolytic enzymes that are going to break down whatever they touch. So here's an example of what that looks like. You have a coated pit that's going to ingest a substance. They're typically substances that have to attach to receptors. Then you have the protein-coated vesicle that's going to detach from the cell membrane and be part of the cell itself. It is now inside of the cell. It can travel around the cell and go through the other side through transcytosis, or it can fuse with the lysosome and become digested if that's what happens there. So phagocytosis is also called cellular eating, and this is how you bring things into a cell. It's something that's used by macrophages and other white blood cells. So essentially, the membrane literally reaches out, surrounds something, and then brings it into the cell in a vesicle. And that's called a phagosome at that point because it has been phagocytosed. It has been brought in through cellular eating. Again, this is common with macrophages and white blood cells. And this happens by amoeboid motion, which is kind of like an extension and contraction. So it's kind of like an inchworm. It'll like stretch itself out and then snap itself up. So that's essentially what amoeboid motion means. It's talking about the movement of an amoeba, which is an extension and contraction phase, to allow the cytoplasm to flow out around whatever it's trying to bring inside into the cell to create a phagosome. Here's an example of what that looks like. You have your receptors on the outside. You have a molecule. So you see that the molecule is not attached to the receptors, but now the receptors in the phagosome are going to be facing the inside. So you know that they were once on the outside of the cell, and this is something that was brought into the cell. Next, we have pinocytosis, which we just talked about. Phagocytosis, which is cellular eating. This is cellular drinking. So this is when we're bringing in large amounts of fluid and whatever solutes are dissolved in those fluids from around our cell's environment. So when that is allowed entry into the cell, it's called a vesicle, and it fuses with the endosome. This allows cells to sample the environment. Essentially, they can determine if there's like nutrients or something that they need for survival that's present in the extracellular fluid that's around them. A lot of times the stuff is the nutrients is absorbed, or again with any vesicular transport, you can have the vesicle be dissolved if it's something that's found to not be helpful. And then of course our membrane components are recycled back to our cell membrane, our phospholipid bilayer, because we're just constantly bringing in things. If it's useful, it's utilized. If it's not, it's hydrolyzed, and then all of the remaining phospholipids, cholesterols, glycolipids, glycoproteins are then returned to the cell membrane. So pinocytosis is called cellular drinking. It's kind of like a gulp of water where it's a large amount compared to like passive transport of water through our membranes. Next, we have receptor-mediated endocytosis, which involves endocytosis and transcytosis of a specific molecule. So a lot of cells have receptors that are on the outside. We have some that are called clathrin-coated pits, and that's just a type of protein. That if something attaches to those coated pits, then it is brought into the cell. So some examples of things that we might want to bring in are like enzymes or LDLs, iron, insulin, things like that. Let's look at a picture of this. So you have those little purple receptors that are on your extracellular surface, and you can see those little like blue teal molecules that attach to those receptors causes like an invagination or like an internal budding of the membrane to create a little circle or a vesicle. And then that is brought into the cell because those receptors are like, hey, I'm looking for my match. Whoever is my match, please come attach to me. And then once that substance attaches to the receptor's like, oh, hey, look, I have what I need. I'm going to start to enter into the cell. And it's like kind of like pulling itself in. So that happens as you have more of these substances attaching to the receptors. They're brought in a little vesicle in order to bring that into the cell. On the flip side, you have exocytosis. So we just talked about endo, which means into, endocytosis. This is exocytosis. So how do you get rid of stuff? Okay, so this is a process where materials are released or ejected from the cell. It's usually activated by cell surface signals or changes in the membrane voltage, which remember has to do with the charges of the ions that are on either side of the cell membrane. So substances being ejected in vesicles are called secretory vesicles. So this is like when you are releasing something in a vesicle instead of bringing it in a vesicle, like vesicular transport that we just talked about. So there's a protein on the vesicles called V-snare, and it finds and hooks up with the target T-snare proteins on the membrane. Don't get too like bogged down with this. You just need to know that you have different proteins that help to drive this function, which is a docking process, and this triggers exocytosis, again, the release of materials. And some substances that you want to be exocytosed or released from your cells would be hormones, because our hormones need to travel around our bodies in order to control other functions that we might do. Neurotransmitters, same thing, mucus, and cellular wastes as well. So here's an example of what that looks like. You have the membrane-bound vesicle migrating to the plasma membrane. So remember that this is for exit. It is leaving. This is the opposite of endocytosis, which we just looked at. Okay, so once it reaches the plasma membrane, the proteins at the vesicle surface, the V snares, are going to bind to the T snares, and they're going to fuse together. So the V and T snares become like intertwined, you can see in that second picture. Then the vesicle and the plasma membrane fuse and a pore opens up. So it's like a slight hole that happens in our membrane to allow all of the materials inside the vesicle to be released to the exterior. And then you have your restored plasma membrane once that flattens back out. This is just like a real picture of what this could look like. So this is a cell actually releasing materials through exocytosis. We're going to talk about something called membrane potential. So this is important also. It's kind of like the gradients that we're talking about that helps to drive the whole process of cellular transport. So resting membrane potential, RPM, it's the electrical potential energy produced by the separation of oppositely charged particles across the membrane. So oppositely charged, positive and negative. So when you have a buildup of a lot of the charges on either side, you have a membrane potential because you now have opposing charges, right? So the difference in the electrical charge between the 2 points is referred to as the voltage. So we're talking about literal charges that are on these various ions. Cells that have a charge are said to be polarized. So we know that from ionic bonds, they're you have polar molecules that have positive and negative ends to them. Same thing. This is a positive or negative end to our cellular membrane. So either the internal or external environment. Voltage occurs only at the membrane surface, so only very close to the surface, either, again, internal or external. Our membrane voltages range from negative 50 to negative 100 millivolts. This is not something you need to really concern yourself with, like what's normal right now. Just understand the whole point of having membrane potential is just saying that you have different ions that exist on either side of the membrane. We're talking about transport, so how things are moving in and out. As you're moving things in and out, especially our ions that have charges on them, like NaCl would be Na plus, Cl minus. Sodium, potassium, they're both positive, right? You're going to have an influx or, a release of our different charges that are going to develop different voltage, different charges on either side of the membrane, and that can drive further reactions from there. So potassium is a key player in our resting membrane potential that we just talked about here. This is where cells are typically like kind of happy. That's why it's called resting. It's not called like sprinting, right? It's our resting membrane potential between negative 50 and negative 100 millivolts. So potassium is going to diffuse out of the cell through the potassium leakage channels, which is down its concentration gradient, which means that is passive transport. Negatively charged proteins cannot leave. As a result, our cytoplasmic side of the cell membrane becomes more negative. So on the interior, you have a strong negative charge and you're releasing positive ions. So you're, getting more and more negative as you're releasing more and more positive molecules to the outside. So when we have our potassium that is then pulled back in by the more negative interior because of its electrical gradient, opposites attract, right? You have your positive charges being pulled back towards the negative side, which is now the cytoplasmic side because we flushed all of our positive charges out. Okay, so that's going back through the electrical gradient because our opposite charges are attracted to each other. So this is when we have the drive for potassium to leave the cell. It's balanced by its drive to stay. So our resting membrane potential is established here. So most cells have an RPM around negative 90, which falls into our scope of between negative 50 millivolts and negative 100 millivolts. An electrochemical gradient of our potassium actually sets the resting membrane potential. So this is all talking about ions and how they're entering and exiting our cells and how that affects the charges on either side. So in many cells, sodium also affects the RPM, obviously, because it's, an ion. It carries a charge. We're talking about charges. That makes sense. So sodium is also attracted to the inside of the cell because of the negative charge that's built up on the inside of the cell. So if sodium enters, this can bring the RPM, resting membrane potential, to negative 70 millivolts, which is still in our normal range between negative 50 and negative 100 millivolts. So the membrane is more permeable to potassium than it is to sodium. So this means that sodium does not travel as easily through the membrane. So potassium is the primary influencer for our resting membrane potential because of this. And then we have our Cl minus that I mentioned earlier. So our chloride ions, it does not influence our RPM because the concentration and electrical gradients are exactly balanced. Also, like it said earlier, you have all of your negatively charged proteins that are on the cytoplasmic side. Those carry a very large negative charge. So like having a couple extra molecules of, a negatively charged chloride ion is not really going to throw the balance off, is essentially what this is saying. So here's an example of what this would look like, and you can see the positive and negative charges. Remember that they stay close to the membrane, okay? Because as things move away from the membrane, like obviously it's not going to have such a pull. So like the positives are not going to be as attracted to the negatives if the negatives exist on the other end of the cell, right? So here, this is going through the generating the resting membrane potential. So you have potassium that's going to diffuse down the concentration gradient through passive transport and leakage channels. And then the loss of the potassium results in a negative charge on the inner surface. So then number two, the potassium also moves into the cell because it's attracted to that negative charge that was just established on the inner membrane face. And then a negative membrane potential of around negative 90 millivolts is established with the movement of potassium out of the cell. And when that's equal to the sodium Nope. Potassium entering and exiting our cell. That establishes our membrane potential. So at that point, the concentration gradient promoting our potassium to exit is exactly the opposite of the electrical gradient for the potassium entry. So the push and pull are equal in that situation. Read through those again. I promise you it's not as difficult as it sounds. Also, the pictures, helpful. So active transport maintains our electrochemical gradients. Obviously, talking about sodium potassium pumps, we're moving ions in and out of the cell. That's obviously going to change the charge, right? So our RPM is maintained through the action of our sodium potassium pumps, which continuously ejects 3 sodiums to the outside of the cell and brings in 2 potassium back inside of our cell. Yes, you need to know 3 and 2. Okay, the steady state is maintained because of the rate of active pumping of our sodium out of the cell is equal to the rate of the sodium diffusing back into the cell. So neurons, our nerves, and our muscle cells, quote, upset this steady state by intentionally opening up sodium potassium channels. And they do this when either you're like flexing or relaxing your muscles, or if a signal needs to be transported along a nerve, right? So then we're going to have an influx or a release of these ions in order to help carry a signal. Cell environment interactions here. So we're talking about how things are brought into and released from cells. So obviously the cell is interacting with its environment. So cells can interact with their environment by responding directly to other cells or indirectly with extracellular chemicals. Because remember, they can bring things in through endocytosis, and they're receiving information about their world around them through these types of like vesicular transport and things like that. Interactions always involve the glycocalyx. This is going to be like sugars that exist around the cell. Cell adhesion molecules called CAMs. It's C-A-M. That's how it's called, a CAM. Okay, and then plasma membrane receptors, which we've been talking about, like receptor-mediated endocytosis and things like this. They're little proteins that exist on the outside of the cells that allow other materials to attach to them like a puzzle piece. They're very specific. The role of our cell adhesion molecules, or CAMs, we're going to talk about here. So every cell has thousands of these little sticky glycoproteins. Glyco is a prefix for sugar. Protein is, a protein. These glycoprotein CAMs projecting from the membrane. So these are going to extend from the outside of the cell. So what do these do, the functions of a CAM? So they anchor the cell to extracellular matrix or to each other. So 2 cells can be anchored to each other with the help of our glycoprotein CAMs. They can assist in the movement of cells one past another. So for like movement of our cells and anchoring. They can also attract white blood cells to injured or infected areas. If you have like MHC receptors that are going to like display different parts of pathogens to be like, hey, white blood cell, please come phagocytose me because I'm infected. They also stimulate the synthesis or degradation of adhesive membrane junctions called tight junctions, which we're going to talk a little bit more about later. And then they can also transmit intracellular signals to direct cell migration, proliferation, and specialization. So this kind of allows cells to communicate with each other in order to coordinate different activities. Okay, so our plasma membrane receptors, these are a little bit different from our CAMs. So what do they do? So they're involved in signaling. So signals are really important because cells can't just walk up to each other and talk. They have to rely on like chemical signals and things like that. So the membrane receptor proteins serve as binding sites for chemical signals. So contact signaling is when we have cells that touch and they're touching each other and they're recognizing each other by the specific surface membrane receptors. This is common with our immune cells. And then you have chemical signaling, which is when we have the interaction between a receptor and a ligand. This is a messenger. It could be like a hormone or a whole bunch of other little materials that can attach to our receptors. Again, very specific, like a lock and key, that are going to cause cellular activities. For instance, in some cells, we have the binding that triggers an enzyme activation or the opening of an ion channel. And some examples of our ligands could be our neurotransmitters, hormones, and some other things that are created around the body. They drive different reactions. So this is how the exterior environment can kind of control the interior environment. By the binding of these to our receptors, it's actually controlling what's happening inside of our cells. And this is a form of communication between our cells, as some cells release things like, hormones, and then those hormones attach to other cells and control what's happening inside of them. So chemical signaling is a really involved process. The same ligand can cause different responses in different cells. So you have one, let's say it's a hormone, one hormone that causes reaction A in this cell can cause reaction B in this cell. It's not always the same response. When you have a ligand that binds, the receptor protein changes shape. It's a conformation change and becomes activated, which can trigger other things to happen. Some activated receptors become enzymes themselves. Remember, enzymes are a catalyst. They speed up chemical reactions. Others can open as an ion gate, which can allow things to suddenly move into the membrane or move out of the membrane that couldn't before unless that gate was open. Those are like our gated channels and things like that. So a whole bunch of different responses can happen because of these little tiny chemical signals. And like I said, they can cause different reactions in different types of cells. We have a chemical signaling revolving around our G proteins. So activated G protein linked receptors indirectly cause cellular changes by activating G proteins, and G proteins help with transmitting signals, essentially, which in turn can affect ion channels, activate other enzymes, or cause a release of internal second messenger chemicals, such as cyclic AMP or calcium. So this is kind of like a cascade event. Yes, that sounds like a lot of words. Well, it is. So there's specific receptors that, when bound to a ligand, can activate G proteins, which are on the internal surface of our cellular membranes. And that's kind of like a domino effect. You knock over one domino and a bunch of other things then happen. That's kind of what's happening here with our G proteins. So essentially, once you have a receptor and a ligand that are bound on the exterior, then that activates our G proteins on the interior, which can then go do some of the things that I just mentioned here. So it's a cascade event. It's a domino effect. Here's an example of what that would look like. Like I said, we have our G proteins on the inside of our membrane, where our receptors are on the external surface. So when you have a ligand that's going to bind to our receptor, the receptor changes shape. The activated receptor binds to the G protein and activates the G protein on the internal side. Then the activated G protein is going to activate or inactivate, depending on the signal, an effector protein by causing it to change shape. Then you have an activated effector. You have an activated effector enzyme that's going to catalyze reactions that produce secondary messengers or messages for the cell. So maybe it's releasing another chemical or something like our cyclic AMP or like calcium. These secondary messengers are going to activate other enzymes. And then you have kinase enzymes that are going to activate other enzymes through like phosphorylation. So as you look at this, you see it's kind of like what I said. It's a domino effect. It's a cascade effect. You have all of these little small reactions that are happening together. So you have the binding of something to the exterior surface that causes this huge chain of events to happen on the interior surface because of our conformation changes and activation of our proteins. And then also lastly with kinases, activation through phosphorylation and things like this. So this is going to be our next section called cellular structures and functions. We just finished our cellular transport. I'm going to stop this here and then I will continue on, and then before your exam, I'll piece together all the different parts. But this is a really big chapter. That's a lot of information that you just received. Please make sure that you read the textbook so it makes sense to you, everything that we just talked about. Please practice the review questions at the end of the chapter that have to do with cellular transport. That's a good place to practice, and sometimes I use those on the exam. Okay, so please read the textbook along with this. We're going to do a lot of practice with this in class, and then we'll move on to our next section here, the cellular structures and functions, which y'all have been doing since like 6th grade. So this shouldn't be as bad as some of the stuff we just talked about. Thanks for listening.